So, uh, glycoconjugation or glycoconjugates is the term given in science in regards to studying or looking at the various carbohydrate attachments and how they're attached and their form and function in regards to their attachment capacities in the animal or model we're examining. Some of the things that carbohydrates actually do within our cellular system is basically work a little bit like a postage stamp and a mailing address. That's the easiest analogy I could provide you in regards to when a particular carbohydrate is presented, and it could go either way in regards to being carbohydrate being presented on a cell surface, and that particular then gives it a particular directional address to go to in regards to whether it's going to the mitochondria or the endoplasmic reticulum, etc. So there's these destination labels for cellular transport. We'll have a look at cellular transport next week, but carbohydrates play a very important role in that in regards to getting the materials to the right place. So, you can call them the FedEx or the UPS of the body. They also allow us, and we've seen a little bit this on the video. So go back to the video of the biology of the cell video. I think I put the web link up there. If not, I'll check and put it up there. Where we see mediation to cell specific interactions by the importance of the cellular matrix. So we saw those leukocytes, those white blood cells, moving along the capillary, and the P selectins come up in regards to indicating a point of infection. And once those P selectins are presented, and I've illustrated this already in some of the lecture notes, that there is a cellular matrix interaction to cause their interlinkage. With that interlinkage, that initiates a whole lot of processes in the white blood cell to give them plasticity to move into the capillary and then into the tissue. So very, very important key elements in regards to the complexity of what we're going to see in the cellular matrix. And again, we'll try and change your view in biology about how complex that particular matrix is. We also have cell-to-cell recognition. So again, carbohydrates can be utilized to recognize self from non-self, and that's very, very important when we look at blood typing. We have also carbohydrates playing a very important role in cell adhesion. So when molecules or cellular organelles need to come together, they might use carbohydrates as a cell-to-cell connector. Cell migration also, as I said, a little bit with the white blood cells in regards to changing their shapes or causing their mobility in regards to, hey, I've got to move over to here. I'm going to express carbohydrates. The signal has been given to me. I'm now going to move within the cytosol to this location. Now, we also looked a little bit at blood clotting, and we'll talk again about some of the aspects in regards to carbohydrates and how they play a very important role in blood cascade clotting. So all these carbohydrates are covalently, not ionically or through hydrogen bonding, they're actually covalently bound to proteins and lipids, which makes them unique in regards to their immobilization in regards to that particular element. So that's why we get the proteoglycans that we saw previously with the bacterial cell wall, glycoproteins, and glycolipids. So this is chapter 7 now. We're on page 261 to 275. So hopefully you're keeping up with the reading every evening. Hopefully you're just chewing a little bit of the reading up to keep up to scratch. So when we think about our cellular wall in our bodies and our cells, again, this is a very simple stylized diagram I got from a biology book. But, the illustration is when we look at the extracellular domain, so this is the outside of the cell, we start seeing all these decorated components interacting with here's a protein being expressed with an alpha helix within the embedment of the lipid membrane. We'll talk about that a little later on. And then there are going to be attachments in specific locations, positionings of these carbohydrates. And these carbohydrates are going to play a role in attracting things, which will then either be attracted or bind to them specifically. And we'll have a look at that actually today. So we have the proteoglycans, or these also called, in the older days, called mucopolysaccharides. Commonly, you might see them in the literature known as GAGs, which are glycoaminoglycans. And basically, these are very large macromolecules which are basically expressed on the as a protein, and then the glucose or a carbohydrate attachment occurs. So they're actually outside of the membrane and kind of immobilized, but a bit like a feather star, if you want to put it in that way. I'm sure you've seen how feather stars, the starfish, they put out those tentacles and they grab their food and then they bring it into the mouth. Very similar process that we'll see with the proteoglycans. The glycoproteins are a little bit more complex. There's usually one or more oligosaccharides, so it could be a very complex, very heterodynamic combination of various carbohydrates. And again, these are also located specifically on plasma membranes, but usually um, inside the cytosolic environment. So remember, you think of the mitochondria, the Golgi apparatus, and vesicles are all going to be moving around within the cytosol. These glycoproteins provide the capacity to identify and recognize, a bit like the postage stamps I talked about before. But there are also ones of glycoprotein nature on the outside of the periplasma membrane, which make up also the extracellular domain or matrix. We also have glycoproteins in blood, actually the red blood cell, which we saw a little bit in the structure with the sickle cell anemia, where the corpuscles themselves, these lovely little disc things, are going to actually be heavily decorated with carbohydrates. And that's what gives you actually your blood type, and we'll have a look at that today. Then we have the other components which are going to be a part of the cellular membrane, particularly is the lipids. And these two also act as targets for carbohydrate attachment. And again, we might see oligosaccharides, these this time being attached to the hydrophobic head of the lipids. So we have that long alkyl chain. So think about our micelles and our lipid membranes we talked about way back. It's amazing to think that we've only done 6 weeks of classwork already, but we've covered a lot of material. So there's these hydrophobic heads that are going to have very specific binding sites and capacity to hold various individual carbohydrates or strands of carbohydrates, which then can be used to decorate the extracellular matrix. But just remember, the extracellular matrix, and I'm going to get back to this picture here, much more complex. So if you think of a forest, and regards the trees going for the sunlight, the small plants, not being able to get the sunlight or be favored with the sunlight to photosynthesize, there's the potential limitation. Same exact thing happens in a extracellular matrix, where the further you can go out and decorate your proteins to attract the ligands, all better for you. You're going to have a better structural form function. You're going to have a higher capacity to be evolved and be expressed. If you're in this particular region where your lipids are containing the glycoprotein, the carbohydrate in the issue of being a glycolipid, there's going to be some situations where your molecule might not get there unless there's a patch where all the lipids are in the same kind of domain, illustrating and presenting those particular carbohydrates as a binding zone. And this is what we call also lipid rafts. And we're going to have a talk about lipid rafts when we look at membranes a little later on. So I want you to keep in member the word of lipid rafts. So here's a very simple stylized illustration of two molecules, a glycoprotein and a protoglycan. Now, the glycoprotein is the easiest one in regards to it's going to be a protein that is going to be expressed, and it's going to basically have a C terminal and an N terminal. But the decorational side in regards to the carbohydrate attachment is only going to actually occur on the cytosolic side. On the extracellular, my mistake, on the extracellular side. There is actually no attachment of carbohydrates on the inside of the cytosol of the glycoprotein. That's going to become a very important feature when we look at membrane-spanning proteins. So note, again, carbohydrates are only presented on glycoproteins on the outside of the extracellular surface of that particular protein. So here we have our membrane, we have the glycoprotein, and you're going to start realizing there can be all these different types of chains in regards to these are stylized kind of code to the type of sugars that are present at these particular positions. But there is something very important here. There is asparagine, serine, and threonine. And they're going to have a look at their chemical functionalities in regards to the kind of carbohydrates they attach, as well as how they attach. So these are attaching to amino acids that are part of the primary sequence of that glycoprotein. When we look at the proteoglycans, they're a little bit more complex because what they do is they still use a protein or peptide backbone, but then they're actually being decorated. So these are actually more like the feather stars in regards to there's these serine moieties within the structure, which provide you the anchor to the membrane. And then they actually have this decorated component in regards to producing a particular type of sugar configuration. And this type of sugar configuration has very specific interactions. So here we have the heparan sulfate and the chondroitin sulfate. Now, this is a reflection of the type of carbohydrates that have come together. And importantly, we know these occur because this little star here, which is xylulose, is the key component of attachment between that and serine. So when we see a serine moiety with xylulose, it's going to be most probably a chondroitin or a heparan sulfate molecule. And again, it is the combination of the various carbohydrates that give you this capacity to do this particular structure. And these heparan and chondroitin sulfates have a particular role in regards to attachment and interactions with other molecules. So let's have a closer look at the proteoglycans. As I said, commonly known as GAGs in the body, and basically they have a core of 30 or so proteins which extend from the extracellular surface to give you the capacity to then give you this decorating component. And it acts like a bit like a brush-like structure. So again, I use the analogy of the feather starfish in its particular role. So the interlinkage of the GAGs have a very specific core requirement in regards to a trisaccharide composition of these two galactoses and this xylulose component. So again, this is what we have here as a clear indication that this is going to be a interactive component of a proteoglycan. After the 5 carbohydrates, so here we have xylulose, galactose, and this is, I think, glucose, acetylated glucose, I think, glucose. Well, I can't remember that one. But what we see here is this requirement to then add to the other part, in this case, the chondroitin sulfate. And it could also be the heparan sulfate. This guy, though, has to be attached by that serine moiety in the primary sequence of the protein structure. So the trisaccharide interlink is coupled to the core protein by what we call is an O-glycosidic bond. And we'll have a look at those O-glycosidic bonds in a second. And so the serine residue becomes that anchor component. What we see in particular is the xylulose reduction, or the reduced end of the xylulose, the alpha-anomeric carbon position, becoming an anomeric carbon to the hydroxyl on the side chain of the serine. So that's why we also see it occurring with glycosylation occurring with threonine. Not necessarily this form of glycosylation, but serine and threonine have those hydroxyl moieties that give you the capacity to bind those particular sugars. So we now have a kind of an idea that there has to be a core protein, a recognition site, a individual carbohydrate in the sense of xylulose being the interactive constituent with the serine, then a number of carbohydrates behind that one, which then give you a little bit of a stem away from the protein backbone, and then a element of repeated components, whether it be heparin or chondroitin, to give you their functionality. In this case, we have galactose, that's the N acetylated form, which also contains sulfur. So again, some very interesting capacity. And it's those sulfur constituents, as we're going to come back, that's what it's called, chondroitin sulfate, because the sulfur is present in that carbohydrate molecule. Do you know what we saw in with the agar? And then we'll see what this does and what this particular molecule can do in our bodies. So let's give us an example of Syndecan, which is basically a large molecular mass of 656 daltons, and it has these very extended chains of chondroitin and heparin sulfate together. So here we have our protein anchor coming through the N and C terminus. So the N terminus is always going to be in the extracellular domain. That's what we call a type 1, but we'll get to those particular protein structures a little later on. And we're going to then have a position where we're going to have the heparin sulfate extending from these particular guys, as well as the chondroitin sulfate being a component at those individual serine moieties. Now, with those extensional components, these provide us attachments. So again, these particular things become very sticky. They can adhere cellular factors, growth factors, and importantly, anticoagulant factors. So this becomes a very important component to capture molecules that might be floating in the extracellular system and that need to be attached to a cell. So again, the heparin sulfate and the chondroitin give you that capacity now to attach those molecules to a cell constituent to then hopefully cause a three-dimensional structural change or some transregulational, transcriptional event or trans-signaling event to occur inside the cytosol of the cell. Now, the heparan sulfate polymer has these S domains, these sulfur domains, obviously, that then bind these extracellular molecules, including drugs. And with that kind of binding, they produce a transductional message through the transformation of the protein structure. So this protein might migrate back into the cell and take that particular factor and motivate or regulate that factor in regards to causing an expressional component. So we see now, in regards to some of these constituents playing a very important role in the issue of combining various heparin and chondroitin sulfate moieties to increase the chemical diversity of these particular types of proteoglycans. We also see this NA, this N-acetylated domain. These are kind of spaces that might be actually present in the heparin sulfate and the chondroitin sulfate. These are not modified or they're unsulfonated, and they can actually then start becoming like a combinational key in regards to how many unsulfonated Na domains do you have compared to how many sulfonated domains. So it becomes like a combinational key. And that combinational key, that spacing component, becomes very vital in how molecules bind to those particular branches. So here we have a look at the heparin sulfate. So here's the backbone of the protein, the sulfur, the xylulose, the three carbohydrates, and we're going to have a look specifically at a region of the heparin sulfate where we're going to see, a gray and a green zone in this particular zoning issue, that this is an Na, an acetylated zone, and this is a highly sulfonated zone called an NS, so an Na and an NS. But when we look at the individual carbohydrate zones, we can see the nomenclature in regards to the number of sulfonated glucose moieties, as well as other glycans and uric acids that are present, in regards to then the stoppage at the N-acetylglycosamine position. So we're going to see that at the stoppage at this point here, and then basically see this heavy sulfonated component with all those negative charges. So remember, the sulfonation is going to give us those negative charges. So obviously it's going to be able to bind something that is positively charged, similar to what we saw with our affinity or ionic exchange ion chromatography columns. So heparan sulfate has this binding region and non-binding region, and depending on the order and the spacing, gives us like a combinational key to allow interactions to occur. Here we see the interactions occurring in regards to a system that I know very well, the antithrombin 3 component, in regards to changing a structural modification based on its interaction with these proteoglycans. So in this case, there's 4 different proteins that interact with this sulfur domain of the heparan sulfate to cause a conformational change. With that conformational change, we then get the attachment of the protein, which acts as an anchor on that NS domain, the sulfonated domain, and then allow you to basically then get the antithrombin component to bind specifically on these individual carbohydrate domains. So we have a conformational change to induce the protein of antithrombin. So here's the antithrombin molecule that's moving around. It's always present, but it's only going to be active once we actually bind it to the NS domain on the heparan sulfate. Once that is bound, it then has a high activity site, which is then going to attract another factor, in this case, factor XA or 10A. In this case, once we get that occurring, we then can get the structural change to actually then bind them together. And with the binding together, the interaction with the blood clotting factor 10A, it prevents clotting to occur. So, if there's an occlusion in regards to or damage to the tissue, the normal situation is that these guys are going to actually form a clot or initiate a clot formation. But with their interaction with the heparan sulfate, they don't form a clot. So there's a structural comparative change that occurs in this particular system. But that's all coordinated by those heparan sulfate domains and how that protein binds, because that negative charge on those heparan sulfates and going to bind to the antithrombin is then going to cause this configurational change based on the charge distribution, which is now being interacted or compensated by binding to the NS domain. So we can see this enhanced protein-protein interaction also with thrombin, which is another part of the blood cascade system. So the antithrombin binding to the domain, as we saw previously here, to the NS domain, causes that configurational structural change. But the change itself does not necessarily only attract factor 10A. It can also attract another protein called thrombin. Now, thrombin, importantly, though, actually has to bind to 2 NS domains to actually do its particular function. So the first one is to find, okay, I will bind to an NS domain here quite happily, but to be stabilized as a protein structure, I need to also bind to the antithrombin constituent, which is also bound to an NS domain. But it means then the spacing component between the non-sulfonated domain has to be very specific, because we could change this and be much further out and not have the capacity to stabilize the thrombin structure, both on the heparan sulfate and its interaction with the antithrombin constituent. So again, this is a multi-phase situation in regards to structural modification and changes, ionic interactions, and also then having this guy initially change due to its binding to the NS domain and not being bound to the factor 10A, but being bound to thrombin. So binding antithrombin and thrombin at two additional adjacent domains brings about a protein coordination. And this is very important because this favoring of interactions gives you the process to inhibit blood clot formation. And again, that allows you to then basically bleed. So those who have hemophilia have problems with this and the antithrombin components. Moving away from another protein-protein interaction with the non-sulfonated and sulfonated domains comes together in regards to this case being a membrane-bound protein. Now, a membrane-bound protein, which we'll see a little bit later on, in this case, this is being a fibroblast growth factor. These are very, very poorly abundant. And for them to find their particular target is going to be actually a needle in a haystack effect, because what there is, there's very little concentration of these, but there's going to be ideally an activational component and a coordination component to get all the receptors together, as well as giving the syndicaine, in this case the NS domain, a coordination capacity, because once it's coordinated, it has a higher probability of finding the ligand to do its job. So here with the fibroblast growth factor, we need to dimerize these two individual proteins. So these might be actually separated in the membrane, and being fluid in the membrane, they can move, but their interactions may not enable them to actually be stable to form the dimer. And the dimer is going to be the active component to actually then accept the fibroblast growth factor. So in this case, given the low concentration of the ligand, it's best to basically try to coordinate all this to increase the molecular probability of interactions and binding. And we do this again with the NS domain. So first off, we get the fibroblast growth factor receptor to form the dimer, come together, and then be there. And that's going to be stabilized by its interaction with a co-receptor coordinator, in this case, the NS domain of the syndicaine. And in this case, once those two elements are together, the NS domain and the dimers of the FGF receptor, then that is active and ready to establish this interaction and attract the actual ligand, in this case, the fibroblast growth factor. With that incurrence, then we get a signal transduction through the membrane and an effect. The idea is binding of these extracellular signals can be limited because of their concentration. It's like an island in the middle of an ocean, particularly with growth factors. And by using these particular peptoglycans, increases the interactions and localizations and increases their capacity to do their particular job. In this case, when binding with syndicaine and basically going through the receptor, we get stimulation to actually cause fibroblast growth and division. So the NS domains interact with both the fibroblast growth factor and its receptor to coordinate, bringing this polymeric complex together and basically increasing the capacity to find these very weak or low concentration, but they're still highly effective. They still have a very high Ka and a very low Kd, but again, just their relative abundance in the extracellular domain is very, very low. So we want to get them, we want to capture them, we want to be able to coordinate those to get the growth factor process to occur. And that is also a process of regulation, because if we don't have this NS domain, or we don't have dimerization of the FGF receptor, we're not going to be able to bind the ligand itself. So again, an illustration of future regulation that we'll see in some cells. We also have this capacity to utilize these with lipids, and in this case, lipoprotein lipases. So these particular guys have the capacity to interact again with the surface membrane because of the negative charges on the heparin sulfate. This, in this case, interacts with these lipoprotein lipases and basically interacts with their S domains to cause a coordination component. It could either have those in a local proximity waiting for an interaction, or allowing them to basically be stored as an active component within the cellular surface. So again, it gives us capacity to localize certain elements using these NS domains. The interesting thing is you might have heard about knockout mice in regards to mice that have capacities to change the normal function and expression of proteins in regards to targeting specific proteins and knocking them out. And how we actually knock those out is actually basically inactivating these S domains. So once we understand the protein form and function and basically being able to knock out those S domains, we have the capacity then to work with an organism that may not be expressing that particular protein of interest, or it can be switched on by an artificial stimulation or an expressional stimulation of these NS domains. So again, depending on how we regulate those NS domains, we're going to get a lot of different effects occurring. Okay, still a little time left. So when we look at our proteoglycan aggregates, as I said, a bit like the feather star component, and the constituents that provide this are these kind of outbreached little areas which have the capacity to bind stuff. And these are very large molecules, many thousands of core proteins bound to the central glycosamine and the hydronic acid containing the glucuronic acid and N-acetylglycosamine basically gives us this little feather star component that basically has this massive surface area in regards to being able to attract and bind to materials. And we can get down to having scanning electron micrographs of these particular proteins, carbohydrates, or these protoglycans, because they're so large. So we have these core proteins, again, the chondroitin sulfate, but then we have this curtan sulfate also in regards to a different component. And as I said to you before, that some of these particular carbohydrates are utilized in regards to structure. We're going to see that when we look at these collagen and fibronectin and elastin, which is going to branch and basically be decorated with this type of material. So again, they give this cartilage and collagen its strength. It's interwoven and basically furthers the strength of the individual constituents. But again, they have these proteins and these sulfonational components that have the capacity to bind certain proteins, including the fibronectin, which binds fibrin, and then basically various capacities to decorate collagen. And this is one of the interesting things about arthritis. If you ever see somebody with severe arthritis where they do an x-ray, you'll see the joints become disfigured and relaying of new bone, which causes actually the disfigurement in the, let's say, the phalanges or the fingers. And that is due to this type of situation where basically the proteoglycans are binding additional materials, which are then inducing an immune response. So we talked about autoimmune responses already in regards to antibody production, where this guy, with its continual layering and movement of bone material, so never think bone is a stagnant component. It's forever being relayered, broken down and relayered. So again, that's kind of illustration what you see with people with severe arthritis and bone disfigurement from arthritis. So this additional binding capacity can have both good and bad components to it. So let's have a look at some of the components that we see in the complexity of the extracellular domain or cellular matrix. And we've already come across our actin filaments. Remember, we've talked about actin and myosin interactions, but actin can also be utilized as a temporal rope or coordinator with different integrals. So integrins were basically these particular proteins that are spanning the membrane, and they're going to then be decorated by the proteoglycans and the fibronectin proteins to give you this large very surface area and this cross collagen fibers to give you the strength. So again, when we think about the strength of the lipid membrane, it's not just the lipids that are playing the role, it's also the extracellular components, which includes the issue of carbohydrates playing a particular role. And when we see carbohydrates in the literature, we might see them in different forms and structures in regards to looking at these types of components where we might see mannose to alpha 1, 2, but this is exactly the same kind of nomenclature that we see in the scientific literature to give you the illustration of the same type of bonding and the same characteristics of a code key that we've seen previously with some of the other constituents in regards to knowing what type of sugar it is, whether it's acetylated, non-acetylated, as we've seen before. Now I'm going to leave it at that in regards to today's lecture, and then we will continue on next week on Tuesday.